Okay, good afternoon. Thank you very much for joining us today. Hi. To this symposium to mark the inauguration of the new Paleogenomics Lab in the Steinhardt Museum. It's a very big day for me, and I'm very excited, as you can see, and I'm really happy, and I'm really happy that you're here today. We have 3 incredible speakers, but first of all, I would like to introduce Professor Tamar Dayan, the chair of the Steinhardt Museum, to say a few words, please. Tamar.
Okay. Hi, everyone, and thanks for joining us today. And we are also very excited. The lab is headed by Dr. Meirav Meiri, and she'll tell you about it. And she and the other speakers, all leaders in the field, will give us a glimpse of the huge promise and huge potential and progress of paleogenomic research. Meirav and this lab are a great addition to the museum as a national research infrastructure. And we're particularly thrilled that Meirav's position allows her both to pursue her own independent academic research, and yet she will be able to provide support for colleagues, archaeologists from all Israeli universities and agencies who wish to adopt this methodology into their research agendas. We know that understanding past human, animal, and plant populations is of great scientific importance, and paleogenomic tools provide an exciting and innovative approach to this endeavor. The museum now has a strong biological archaeology research program with paleogenomics, archaeozoology, archaeobotany, and palynology, archaeomalacology, all with a great cooperation with the Department of Archaeology. And in the Dan David Center for Human Evolution and Biohistory Research, Physical and Dental Anthropology and Ancient Human DNA. So we've been given the great opportunity for systematists and biogeographers, anthropologists, and archaeologists to interact closely, and we're positive that this new lab will be an important contribution to the development of this multidisciplinary research framework for all of us. Credits for the Paleogenomics Lab are due, and I'm particularly happy to give them today, and I'll read them carefully. First, this lab was established in an earlier format by Professor Israel Finkelstein of the Department of Archaeology at Tel Aviv University. As you will hear, paleogenomic research can provide exciting new insight into time-honored questions at the core of archaeological and paleoecological research. And a decade ago, Israel Finkelstein had the vision of its potential contribution to archaeological research in Israel, and he also led the submission of the grant proposal to establish this brand new and exciting lab. The Ancient DNA program in the lab is overseen by a steering committee that includes Professor Finkelstein, Professor Dorothée Huchon from the Tel Aviv University School of Zoology and the museum, Professor Guy Bar-Oz of the University of Haifa, and Professor Liran Carmel of the Hebrew University. Their ongoing partnership and support have been and remain incredibly important to the success of this endeavor. Yad HaNadiv, the Rothschild Foundation, supported the establishment of the lab and that of the associated research grants program, and the remainder of the required funds were provided by Mr. Michael Steinhardt of New York and of the Steinhardt Museum, and we are deeply indebted to all. With these colleagues and with these partners, we look forward with optimism. So we thank the symposium speakers for their willingness to share their time, their knowledge, and their expertise with us. And again, thanks all for joining us today. We wish Meirav much success and all of us an exciting and illuminating symposium. Thank you.
Thank you, Tamar. Now I'm pleased to introduce Professor Yuval Godot, the head of the Department of Archaeology and Ancient Near Eastern Culture at Tel Aviv University. Yuval, please.
Thank you, Merav. Thank you, Tamar, for your kind words before for our department and for inviting me to greet the participants of this highly interesting online symposium. It's good that it's online because we have people from all over the world, not just because of the corona. The inauguration of the Steinhardt Museum Paleogenomics Laboratory is a very blissful moment for itself as we celebrate the achievements of everyone who is involved. However, it is also a milestone or a landmark, if you wish, along a very long road that's literally being cleared out and created by Merav, Tamar, and their many partners. I have to admit and say that I am a witness for this continuous effort for over a decade, I think even more now. And so it's very clear to me that nothing of what we are seeing and what we know about this museum and its labs can be taken for granted. The archaeology department here at Tel Aviv University has been enjoying the fruits of this center for years now, as we were able to intertwine and establish interdisciplinary labs for archaeozoology, archaeobotany, and archaeomycology. And thanks to that, to expand our research, academic staff, and our curriculum. We are grateful for this, and we know that the centers like the one we are all gathered here to celebrate, its inaugurations, are centers dedicated to the creation of knowledge, and so are at the forefront of science today. Thank you again, and I hope you will all enjoy the lectures today.
Thank you, Yuval. And now this is my part. Second, okay. Okay. Can you see the presentation? Okay, cool. So as I said before, it's a very big day for me. And in the next few slides, I would like to show you the lab and a little bit about the work that we are doing here. But first of all, where we come from. So we had a little ancient DNA lab in the Department of Archaeology at Tel Aviv University. And you can see from the picture that the surrounding is very adequate to the kind of work that we're doing. But unfortunately, the lab was very small. Only one person can work there at a time, and it was not well equipped. And by a lot of efforts and work by Professor Israel Finkelstein and Professor Tamar Dayan, we received a grant from Yad Nadiv to build a new lab. The new lab is in the beautiful museum. And I have to say that the building of the lab was much more complicated than we thought before. So I think it was the combination of the location of the lab with the lab requirements that make it very challenging for the contractor to build it. And you can add to that the COVID, and then I haven't been here for a year. So you can guess that it took a lot of time for the lab to be finished. But in the end, I think it's worth it. It was worthwhile waiting for it. And the new lab is big and it's beautiful. And we have a room for sampling and the main room where we do the DNA extractions and preparing for next generation sequencing. And it is really, really great. And I'm really happy. Now, a little bit about the work that we do. So we did a lot of work on DNA of pigs. And we found out that pigs are excellent proxies for reconstructing human migrations. And when I say we, I mean a big team of really excellent collaborators. And mainly, I mean Israel Finkelstein. And what we think is that domestic pigs are relatively small and they have high percentage of fat and they are omnivores, so they are not picky with the food they are eating. And that makes them good candidates for traveling. And you can think about them as a complete lunchbox that you take with you. It's very convenient. And together with the fact that they have a phylogeographic structure patterns that across their distribution that we can follow, and we can distinguish between European and Near Eastern pigs that help us to follow their path. And what we found was that domestic European pigs were brought to the southern Levant during the Iron Age, and we think even earlier, but this is not the only direction. We found evidence for, for pigs from Near Eastern that were brought to Sicily and Sardinia, that you can see on this map. And we can also see Italian pigs that came to Greece during the late Bronze. So people are moving and they are taking the pigs with them. And from that, we can learn about trades, migrations, and even culinary preferences. And pigs are interesting. And they're even more interesting in our region because we found something really unique in the modern Israeli wild boars. So if you look only on the mitochondrial DNA, the wild boars from Israel fall within the European clade. But if you look on the SNP data, they fall in the Near Eastern clade. So we have this some strange of a hybrid, and we don't know exactly when and how this situation came about. And this is something that we need to keep explore, but we are sure that domestic pigs were brought from Europe to this region, most likely several times during the course of history. And some of them went feral, ran away, and they interbreed with the local wild boars. We also worked on cattle, and we found an evidence for hybridization between a cattle and a zebu in the Levant. And it seems that the ancient farmers, they knew what they're doing. And by the crossbreeding, they created a cow that is more adapted to dry climate. The zebu, which originates in India, has better heat tolerance and has a better resistance to tick and insects. And in the light of archaeological and historical evidence, we believe that Egyptians during the Bronze Age, this is an attempt by them to expand farming in a region in a period where there were severe droughts. Now, a complete different project that I'm working on together with Professor Guybal Oz is about grapevines from the Negev highlands during the Byzantine time. Now, there is a lot of great researchers that are working on this project as well. And the thing is that during the Byzantine time, in the Negev highlands, the people in the region established a sustainable and a very sophisticated land agriculture. And one of their main achievements was the development of intensive wine production. So they used to grow the grapevines in the area. And we think that they may have some adaptive characters to this arid region. And then they used it for the wine production. Unfortunately, all the knowledge about the varieties that they used and the wine production was lost because of the Islamic conquest. And the aim of this project is to reveal the lost knowledge of the Byzantine grapevines. Now, I'm not going to show you results on this project today. This is an ongoing work and we're still analyzing the data. So to be continued, let's talk in a year's time. And another project that I'm working on belongs to Dr. Nimrod Marom. And we focus on the Judean desert and specifically on the remain in caves located there. Now, the aim of this project is to explore the effects of human settlement on mammalian community, focusing on the trophic cascades. Now, again, we just started to work on the material here, so we don't have a lot of results, but we have one surprising findings. And one of the findings was assemblage of bones that belong to the crested rat. And they are dated to the late Pleistocene, so around 40,000 years. And that was a great surprise to find a crested rat in the Judean desert. And the modern distribution of this rat now is restricted to East Africa. And this is an incredible rat. It's a poisonous rat. It uses the poisons from a bark of a specific tree and it just smears it on the hair. And this is how it defends itself. And we managed to extract almost a complete mitochondrial DNA from one of the specimens. And this indicates a good preservation from this site. And hopefully this is a good sign for the future. So cross your fingers and we hope that we will have good results in this project. And another thing is that we recently distributed a call for research proposal on ancient DNA. So if you have a project and if you are interested, the deadline is the end of June. And this is the time to submit. And the lab is here in the museum and it's open for collaboration. And that is it from me. That's my end. And I hope now you're ready for to hear some fantastic talks. So the first speaker, I will just stop sharing the screen. Okay, so the first speaker, I'm pleased to introduce Professor Tom Gilbert from University of Copenhagen. And Tom will talk about temporal genomics as a tool for conservation. Tom, thank you.
Thank you, Mirav, and thank you everybody for having me here today. It's really exciting to see the lab ready to be opened. I've been following what Mirav's been up to since she was a PhD student with Ian, which feels like a very long time ago. And it's really exciting to see this. And it's also nice, I was just looking at the audience, I recognize a lot of people in there. I saw Guy and Nimrod and Eli and things, so people I work with. So thank you for attending. So what I am going to talk about is about how we can maybe start to use time series of samples for applied conservation. It's not a topic I used to work on, but I've become more interested in it, and I apologize if I say anything very stupid. Also, my connection at home is a bit funny, so Mirav, please interrupt me or something if I go down. But I'm based in Copenhagen at the Globe Institute, which is we used to be part of the Natural History Museum, but we were rearranged, and I'm also partly in Trondheim in Norway. So the reason I got into this topic was because about 3-4 years ago, I got an email from this man, Damien Fordham, who's at Adelaide, and he was holding an IUCN workshop, and he said to me, could I please give a presentation on evidence for the effects of past climate change or anthropogenic activity, so essentially the environment, on genetic diversity? And I immediately said, sure, that sounds super, super easy. And of course, you know, one could ask, why might the IUCN care about this? Well, of course, the IUCN is the International Union for Conservation of Nature, and conservation biologists love genetics these days, and they are obsessed with genetic diversity. And ideally, there is a hope that genetic analyses, like the kind of things we're all doing, could or at least should inform conservation efforts to maximize diversity, because there is this idea that genetic diversity is one of the key things we have to preserve out there. So I was thrilled to be asked, because as I said, I thought this would be super, super easy, because obviously the answer is yes. Of course, like past processes do affect genetic diversity. So I was like, this will be really, really easy. And then I forgot about it. And then when I got round to writing the talk a bit later, like 9 months later, I thought more about it. And my assumption for saying yes was, well, it's got to be yes, because we know that the environment definitely changes population sizes, and it definitely changes population structures and connectivity, and it definitely drives selection, and all of these must affect genetic diversity. So super, I'll give a talk and it'll be easy. The problem was, when I actually read his email carefully, just before writing the talk the day before, or whenever it was, of course, there was this word in yellow, evidence. And this is when I just got slightly worried, because things that are intuitive may not be that easy to support based on evidence. And so when I thought about it, well, obviously, like, you know, there's a lot of theory that says, yes, it's the case. It's easy to do models and show that when you change things, diversity changes. And there's lots of good laboratory evidence. I'm sure many of you have heard about the fruit fly experiments that people have done for decades and decades, where they basically keep fruit flies in tubes, and every generation they enforce bottlenecks or change the population and watch what happens. And clearly, yes, when you play around with them, things happen. But actually, when I started looking into trying to find good evidence in nature, it wasn't quite so straightforward. And you can then ask, why would this be? Well, firstly, theory and models that people use may not actually recreate the complexity of life. I mean, by necessity, we have to oversimplify things. And actually, the real problem is most people studying this question are looking at modern data sets, right? They will sample a load of modern individuals of a population, and they'll see a pattern, and then they have to invoke an awful lot of assumptions to make sense of what they see, and assumptions are always a slight problem. So does genetic diversity matter? Well, as I said, a lot of very influential people say yes, and again, there are observations that support yes. I mean, we know that severe inbreeding is bad. The classic example most people in Europe know about is the Habsburg monarchs. These guys were so inbred that it's still causing problems in the monarchies of Europe today. If you have these super inbred populations, yes, you have a problem. And you can also see this with the French Quebecois and other communities. But I was more interested, you know, well, let's look in nature and see what's going on. So here's one example of where we clearly can see something bad is happening based upon modern samples. And this is the Crested Ibis, which is a bird I became familiar with a few years ago, and it's quite an interesting story. So if you look in the figure A here, you can see its range in the past, and in green is the range today. It's very restricted, but what was interesting about it was, if you look in figure B, the populations basically collapsed over the last 150 years, and actually in 1980 they were declared extinct. There was a couple of them left in Japan that died out, and that was the end of the Crested Ibis. But in 1981, actually a group in China discovered 2 breeding pairs, and suddenly we had them back. But this is a species that's been through a crazy bottleneck, right? They've been down to 2 pairs. And now there's about, I think, 2,000 individuals. They are very intensively managed today by I guess, Chinese people. They basically hand raise them, and they're looking after them. And so they're getting a lot of work. But it's a very good example of an extreme bottleneck. And when you actually look at the species that's been through this bottleneck, I mean, they're not doing very well. If you look at their wild deaths, I mean, a lot of them die, like nearly 50% died from disease. And this is disease either because their immune system has lost all its diversity or because it's genetic diseases. So they are clearly in trouble. So that suggests that, yes, diversity is a problem. But the problem is, there are lots of other studies that disagree with this. So here's a study I like. This is the island foxes. These are from the Channel Islands off the coast of California. They're these tiny foxes, which are apparently very tame and at risk of getting squished by hikers or something. And the nice thing about this study, it was looking at the genetic diversity of these foxes, which are doing completely fine. There is no evidence really they're in trouble. And if you look at this figure here, each of the stars is the genetic diversity of one of these island foxes. That is crazy low. Marked onto the histogram are various other species that range from species that we know are in trouble, A and B, like the Yangtze River dolphin and the cheetah, through to species that are doing all right, like zebrafish and stuff at the other end. And if we consider my Crested Ibis, that's point D, you can see here on the left. These went down, and say, to a bottleneck of 2 breeding pairs, and they're very screwed up and they have low diversity. But these Channel Island foxes, which have got way less diversity, are doing completely fine. So it's not straightforward to say that low diversity is a problem, right? There has to be more to it than that. And this is where studying ancient samples gets maybe a little bit interesting. Because in light of these kind of observations that the story is not so straightforward, it raises questions that should be tested, that are questions ancient DNA can help. And they're things like, does theory actually replicate reality? So if we model something and then we actually look at something, do we see the same thing or not? Which is kind of important if you're going to make lots of decisions based on the output of theory. If we see patterns, is it correlation or is it causation, right? And this is the problem with modern data sets. It's very easy to assign causation to correlations, and that's not ideally the best thing. And actually, are genomic measures of anything actually useful for conservation? And if so, which ones are they? Is it just diversity, or is it more advanced things like runs of Hemorrhagicity or so on? And of course, we do need to know all this, because from the conservation point of view, if we're seeing population crashes, we have to know, are they actually going to cause problematic levels of low diversity? Or will they just cause low diversity that's not a problem? Or will they not cause diversity to decrease at all? Another important point, how much diversity do you actually have to lose before there is serious consequences? And probably, how do you have to condition this on the taxonomic group you're looking at to start with? And indeed, are some of the observations we see today actually unusual? For example, rhinos have got very, very low diversity, which may be a cause for panic, unless suddenly you can look back through time and see they always had low diversity, then maybe it's less of a thing. So this time information is super important. That is not to say that it can't be done with modern data, and I do want to make this very, very clear. And you know, there is a lot of people out there sequencing genome scale data sets right now from modern populations. I'll just give one example. It's the muskox, which I've sort of played around with every now and then. There's me actually doing fieldwork, which is extremely rare. And I'm actually just showing off while Anders behind me is doing the fieldwork. Now, the muskox is kind of, you know, it is a good case study. This is projections, we're modeling projections, hindcasting projections of their range through time. This was done actually for another study about 10 years ago, looking at the fossil remains and modeling the environment. And we can see as we go from left to right, as we go from 40,000 years ago to 6,000 ago, the range has changed a lot, right? So we know these range changes have happened. It must have done something. And if you sequence the modern genome and you do some kind of population history reconstruction using PSMC or MCMC, these kind of things, and that's what this figure shows here, we can see that as we go from right to left, from 50,000 years to the present, we can see their population size going down, so it kind of fits the range changing. And if we break up the population today into subpopulations, for example, Canadian populations on the left and Greenland on the right, we can see variable levels of diversity. And you can dig into this and come up with all sorts of explanations for why that is. But the problem is, it's all the explanations we're coming up with are based on many, many assumptions. So we're assuming, for example, in Greenland, there is very low diversity because they only entered Greenland 4,000 years ago, and that might have caused it. But similarly, they may have low diversity because the population they came from 4,000 years ago had low diversity. So these assumptions are problematic. And this, again, is why I mentioned going into the past would be really awesome, right? Time travel. And this, of course, is the promise of ancient DNA. And this is why it's great to see labs opening today. I hope your lab is as fancy as this one, Mary. I don't know which one this is. This is my favorite one on the internet. And one day someone will explain to me what the glowing boxes do for ancient DNA. But I like to show. That's what an ancient lab should look like, glowing boxes. Doesn't matter that nothing's actually happening in there. So ancient DNA, of course, has been used to look at species through the past. When I started my PhD in Oxford back in 2000, there was a wise old postdoc in the lab called Ian Barnes, who luckily we'll talk later. And Ian was actually studying mitochondrial DNA on bears just to look at what happened in the past. And Beth Shapiro was doing it on muskox. And when I left Oxford, I got involved in a study on muskox, again, using ancient mitochondrial DNA, and we basically built some big phylogenies. You can see this on the left, and color basically tells us where things are coming from. But on the right is more interesting. This is a skyline plot showing the population sizes of the different clusters of muskox going from 80,000 years in the past to zero, going right to left here. And we could reconstruct the populations for going up or down. And what we typically did with this data was then try and overlay interesting information on it to explain what was going on. So marked on this figure is various marine isotope stages, and the key point is things like MIS2 here. This is when it's getting very, very cold, and we can see the population of muskox was going up, and we could say from this, well, that's awesome. We think that when it gets cold, the muskox's range expands, and they expand. So this stuff can be done, but the problem is, of course, it's mitochondrial DNA, and mitochondria is useful, but how representative is it really of the nuclear genome? And because does it really tell us anything about what's going on in the nuclear genome? And if the nuclear genome is actually doing the important things, encoding for genes that do stuff in immunity, will we ever get where we want to get on the mitochondrial DNA? And now, luckily, of course, we have the right technologies in place and we have the right money in place, or more importantly, actually, the cost is going down of doing this. Paleogenomics is kind of the way that many people are trying to go forward, and one has to give Svante Pääbo really the credit for kicking this all off about 20 years ago. But it's really getting very, very affordable now. I mean, things that the amounts of data we can generate today mean that for many, many, many samples, it is feasible to get into the genome scale data. Now, Luva Darlin first did this, really, I think, on the animal context. He published a paper back in 2015. I love this paper because it's based on 2 samples, but actually it's super interesting. As many know, Luva is a mammoth man. He's very into his mammoth. And what Luva did in this study to showcase the potential, really, of ancient paleogenomics, he was looking at mammoth extinction, and he had 2 mammoth samples. In pink on this map, you can see an Oymyakon mammoth 45,000 years ago. And on Wrangell Island, up in the blue there, was one of the really last mammoths, the ones from like 4,000 years ago, which is kind of crazy. People tend to forget mammoths were around when Egyptians were building pyramids and stuff. And they were around in these tiny relic populations, pretty isolated, because area that had been land, as shown by the red dots here, was becoming more and more isolated as the ice melted and the sea levels rose and so on. So at about 4,000 years ago, there was this relict population on land that was getting shorter and shorter and shorter, so smaller and smaller. Now, what Luva did and his team, they sequenced the genomes of these 2 mammoths, and they then did various analyses to look at how is the genome changing through time. And that's the key thing for really understanding what's happening to species, actually looking at what is going on. So what we can see in the bottom right here is a PSMC plot, just showing basically the population size of the Oymyakon and the Wrangell mammoth in pink and blue through time, going from starting like 4 million years ago on the right through to 4,000 years ago on the left. And we can see that they show very, very similar trajectories for most of this. In fact, until the Oymyakon mammoth dies at 40,000 years ago, which is good. It's showing it's one big continuous population doing the same thing. And then what we can see is that as the Pleistocene-Holocene transition comes in, bam, it collapses down, right? So really, really good. We see that loss. And then what we can do, or what Luva could do, was dig into the actual data and look at various measures that might be important for conservation. So, for example, heterozygosity, shown here on the left, just how much diversity is in the genome. And what you can see here, if you look in the middle where the arrow is, that the Oymyakon woolly mammoth, it had a certain amount of diversity, and then the Wrangell mammoth, 40,000 years later, it had changed quite a bit. And so we can actually see, yes, as the population is going down, we are losing that heterozygosity, which is what we would think would happen, but it's kind of nice to see that. It doesn't mean that actually the loss of heterozygosity was a problem for it. That's another question, but at least we can measure it. And another thing they could show, they were looking at runs of homozygosity, lengths and frequencies. So these are tracks of the genome with no variation in that at all. And basically what you can see if we look in this zoomed in bit here is that there was basically not very many runs of deserts of variation in the Oymyakon one. But when we look at Wrangell, wow, it's been purged across the genome. It's getting more and more and more clonal looking, right? And actually being able to measure this just on 2 is interesting. And of course, what then gets really interesting is adding samples on top of this to really look at how the process happens and how fast does it happen and what is being affected by it. So the good thing about Luva's study, it basically showed that it does work. But he is limited by the fact he was looking at mammoths, which aren't around to bring back anyway, really, today. And there's not a particularly good reference genome at the time for that. But it showed the potential, and the potential that if we do start to have very good reference genomes for species, and we can get access to many well-documented modern and historic samples, and well-documented as in location and age of them, and even better, if we have a realistic idea of what actually happened to them, so we can actually calibrate what's going on based on evidence as opposed to guesswork, which is what we have to do in things in the deep past, then we can really get some powerful insights. And so I'm going to give now for the rest of the talk just 2 examples of the kind of insights you can get. The first one was a project I did in collaboration with Guoji Zhang here, who is kind of at BGI in China. He's also in Copenhagen. And also collaborators in Barcelona. And Guoji and I have worked on bird genomics for quite a long time. And Guoji and I were leading this effort to sequence many, many modern bird genomes. And actually, we even have samples from Israel for that, thanks to Tali Magari, who was there, is now with us. But as I mentioned, there was already this pre-existing paper on the Crested Ibis looking at the modern individuals and what happened after the bottleneck. And so what we started to do was sequence them before the bottleneck to see really, how much of a change was there really? Did they lose enormous amounts of diversity, or basically were they screwed up beforehand anyway? So we went around collecting historic Crested Ibis samples. And in doing so, we could basically very much expand upon where they come from. So today they live where this pink cluster is on the map here. But as I showed in the earlier part of the talk, they used to basically live all across East Asia. And by sampling from various museums around the place, we could basically cover a decent part of their range, including places like Korea and Japan and all over China. Once we sequenced the genomes, and this was from foot pad material from samples that were about anywhere from 100 and 200 years to sort of 1910. So not particularly old, but from good historical dated provenance material. We could do several things. So for example, we could build a phylogeny on the genomes, and all you need to see here really is the modern ones fall into this little pink group. And in the past, there was a lot of structure and a lot of diversity that was lost. This is telling us straight off, yes, okay, we have had a catastrophic loss of both diversity and structure, right? Which you could assume, but having said that, when we knew the range was just a big, large area in the past, that could have been a big panmitic population, right? But this is showing, no, that there appears to be quite good structure. And this is also mirrored in the principal component analysis on the right. But the other thing that we did, I thought was quite interesting, was we could run methods like PSMC that allow you to take a single genome and look at the population size through time. But we could look at it in the population-specific context. Now, what I'm showing in the left here is our estimate of the population history of the Crested Ibis having sequenced one of the modern genomes, right, from the remnant population. And we did this in the first paper, the one from 2016 or whenever it was. And what it showed at that time was that the population was kind of healthy until about 1,000 years ago, and then it really, really crashed. And at the time, we used this to say, well, like 1,000 years ago, there's real intensification in the agriculture and so on in China and the regions where these are found. We're pretty sure humans were to blame for this collapse, right? But when we replicate this analysis using the different subpopulations, we see some really quite different stories. So yes, in the Northwest, we see something quite similar. But if you look in the East Asia, there is a drop, but it's much less catastrophic. And if you actually look in the Northeastern things, I mean, the drop basically didn't happen until very, very recently, right? So it wasn't this interaction with humans for a few thousand years. It was really humans in the last century that killed this thing off. It's extremely clear from just breaking up the data into these populations. And this highlights the problem of studying relic populations today. You lose all this prior information that can very much color your understanding of what was going on. Another thing we could do is just measure genetic diversity. So basically the pink line here is the genetic diversity across the genome of the modern ones today, and the blue line is the ones across, it's the average across the ancient ones. And so again, we can start to quantify the loss of diversity. This doesn't necessarily mean loss of diversity was a problem. That's what one can start doing next by digging into where is the diversity lost, and are we seeing increases of loss of function mutations, for example, or deleterious mutations and so on. But in summary, what the Crested Ibis data really showed us was, yes, they're in trouble today, but they looked pretty healthy until the late 19th century. So it was very much a recent reason for the collapse, right? But also these different populations across the range did have very different responses to the environment in general, right? So one has to kind of factor that in. You know, some of these populations were suffering for much longer, and one can then maybe start to look into why was that versus why weren't other ones doing that? Was it just range change, or was it actually they had sort of entered some kind of extinction vortex thing where their genomes got into trouble? I don't know what it is, but one can start to ask these questions. Definitely the climate probably wasn't all to blame, and so clearly, you know, humans are the bad players in this one. So this we found very encouraging, and then we started to think, well, you know, what can we do about this? Well, basically we can avoid past mistakes, but I do know people in this crazy revive and restore community who are starting to talk about, can we use this information to sort of bring things back? Could we, for example, look at the variation in the lost clades of Crested Ibis And if we can identify important stuff, can we actually edit that into the genomes of the living ones and bring back key mutations? I'm not saying it's a good idea, but it's the kind of thing they want to do on these data sets, right? But again, it does raise the question of how representative this is, and this is why more and more studies need to be done, and this is why it's great that more and more paleogenome labs are starting to appear, because as I mentioned earlier, we're in a situation where there are more and more good reference genomes coming, and by good I mean well sequenced and well annotated, and there are more and more data sets being documented online, and this is a big problem, of course. One can decide to do a study on a specimen, but if you can't find out what material is there because it's in a museum where it's not documented, not much is going to happen. But as more collections get documented, more and more potential comes. And so I want to end with just one more example of the kind of interesting things you can start to see. And this was a project we started about 10 years ago. Ross Barnett, who's on the right of the picture here, was a postdoc on a Marie Curie grant. He's obsessed with lions, and he started a project and didn't finish it. But then Mark Montero, who's on the left here, was in Barcelona, was very interested in finishing it. So together they studied. And what we did was we looked at the genomic history of lions through time, and not just modern lions, but we also managed to get our hands on some cave lions, the Panthera spelaea and atrox, which are basically modern lions, but subspecies of them. And so what we did in the study, we tried to sample lions across the historic range, and basically the little shapes here are some of the historic samples and the modern samples. It wasn't a huge number of samples, it was like 16 or 17, but it did cover the key regions. It covered the Barbary lions up on the left here, which have gone now, the sort of Mesopotamian lions, which are gone now, the Indian lions, which are still here but in big trouble, and then various other populations where they are either doing okay, for example, down in the south, or in big trouble in the west. And on top of that, we had a few cave lions actually from high up in Siberia and the Canadian Arctic. So we sequenced the genomes just to start and look at what was going on, and we could see several things. First of all, we could see that yes, the cave lions were pretty unique. Having said that, our cave lions are from a long way away. There were cave lions in Europe and places, and maybe they were closer. And we could see in the phylogeny there is structure, but there is clearly this big split. You can see in the middle here these two clades that basically separate lions from this part of Africa, the range today, and then lions from the north. But the interesting thing is these west ones from Senegal cluster with the north ones. So the split is basically south and east, and then north and west. Now, why might that be interesting? Well, if we start talking about replacing lions or bringing in lions to replace lions in trouble, you need this kind of information. I'll come back to that in a minute. We could also actually see a few useful things for the conservation point of view. So for example, the Barbary lions, as we can see up here, they did form their own clade, and there are people arguing they were a distinct thing and something should be done. Well, we can see they were a distinct thing, but actually down in the south, we had the Cape lion. The Cape lion is sitting in the tree here, and there's two Cape lions. These are described as a distinct subspecies, but they're clearly they're not monophyletic, at least based upon the data today. So with this information in mind, that may color people's ideas about which ones might be worth trying to put effort into restoring or not. Were they unique or not? So that was useful. We could also look at the divergence. We could use various techniques for doing that. I'm not going to go into how we did that, but basically we could see that the divergence is not great. We're talking in the context of tens of thousands of years for the divergence between basically the southern clade and the northern clade. And I will get back to that because that is useful in a minute. And the divergence with cave lions is in the range of 100,000 plus years. Not a huge amount, but not recent. But it's not a huge divergence between the southern ones and the northern ones today. So bear that in mind. And of course, the divergence when you get down to it between, for example, the Barbary lions and the West African lions is even less. We could also see, and that's what the figure on the right shows, there was a lot of admixture going on. Basically, all of the extant lions have got admixture with the other groups in their past. And this includes things like the Asian lions today. They weren't isolated for a long time. They've got a lot of gene flow from the lions coming from at least West Africa. And this is really important information when it gets to conservation. What we could also do, we could split them up into populations and actually look at the diversity loss. Because we had samples from 100 and whatever years ago and the present, we could not just measure the diversity today, but we can measure it then and actually measure the loss. And there are different ways of basically viewing this. You can, for example, just view in the top right here, the the diversity across the genome of different populations today. So this is a Tanzanian lion. In orange, you can see it's got a lot of diversity all across the genome, whereas the Indian lions have lost a lot of it. You can actually look at it as shown in the bottom figure, which shows the lengths of homozygous DNA in the genomes. And generally, the more inbred you are, the longer they are. And we can see that basically, yeah, the Indian ones are really, really screwed up there, but other populations are doing all right. And this is interesting. Senegal, for example, there are basically very few lions left in Senegal and West Africa. But diversity-wise, they're doing great, which is, again, very counterintuitive, right? We would imagine they'd be like the Indian lions, which are very, very restricted. But what I really like is actually showing it this way. What this shows is the genome of a South African lion, and basically the top left is just the start of the first chromosome, and the bottom right is the end of the last chromosome. So just reading left to right is zooming along the chromosomes. And green dots are heterozygosity, red is basically there's no diversity. So as you zoom along the chromosome of the African lion, you see diversity, and then you see, okay, bits where there's none, and then a bit more and bits of none. This is a South African lion today. This is one 100 years ago, and this is where you can really start to see big change. Same population, 100 years. This is not many generations. Huge tracks appearing, right? So this is the really powerful insights you can get on really not very old material. And this South African lion is from a hide, right, that was just preserved. I mean, your collections are full of these. And actually, I mean, this is extremely striking. Again, it doesn't mean it's actually functionally important, but you can start to measure this very well. This is an Asian present-day lion. Asian lions, Indian lions are in deep trouble, and you can see why. They're basically monozygotic twins. I mean, it's crazy, crazy, crazy kind of stuff. And actually, when you look at the Barbary lion as well, you see a different pattern. And it's very, very clear when you dig into the data that what you see correlates with pressure from human civilization. South Africa, there was not a lot of pressure from humans really until, whatever, 200-300 years ago. And it's only really become a problem in the last 100 years. The Barbary lions, which were basically bang in the range of the Greek and the Roman and the Egyptian civilizations, they did show a lot more problems, more like the South African present-day lions when we looked at them 100 years ago, because they'd had that pressure for longer. And of course, India, where there's been a lot of pressure for a long time, really, really deep trouble. What though we could also do, and I think this is the most interesting thing, this is the Siberian cave lion. Now, this thing is 35,000 years old. You can't blame humans for everything. And the nice thing about having things like this, they give you a nice baseline about how things did look before humans come. And when you compare that to the South African lion, it's pretty striking that basically not much had happened, right? It really does show that lions were doing fine, at least in Southern Africa, because they were looking like the default state. And it's very nice to have these benchmarks, because they do allow you to start to see what is a problem. Had I just looked at this South African 120 years ago without this, I wouldn't have got any idea of, had it already suffered and lost some diversity, what was it looking? And more studies are starting to do this now by having true unaffected samples, which is when the real paleogenomic samples come in. So if we conclude on the lions, basically, yes, we can see that some of them are genetically depauperated, and how badly that is correlates with time of overlap with civilization, but it is definitely getting worse in the past century. Indian lions are in huge trouble. You don't need genetics to tell you that. They have problems with like sperm motility and so on. But the interesting thing, going back to the admixture analysis or the analysis of looking at mixing, I mean, we can see that these Indian lions are not pure anyway. We can see that until recently they were getting gene flow from, well, based on our data, the Barbary lions and clearly the Mesopotamian lions, if these start to appear, and even the West African lions. And this starts to raise very unpopular ideas like, well, maybe we should consider introducing West African lions into India. Now, I'm not going to take that argument. There's huge amounts of politics against it, but at least we can see in the data, gene flow is not unusual for these. So if you ignore the politics and national pride and blah, 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 at least there was admixture suggesting there could be a reason to mix it. However, on the other hand, there's basically, you know, less admixture between other groups in other species, and then one can use that to condition the conservation things that are going on. So what does this say might be done with lions? Well, you know, if we want to restock the Cape and the Barbary populations, that could be done. We could just take regular South African lions and stock them in the Cape regions, because that's all they were. Barbary lions were unique, but not that unique. So if we moved in West African lions there, we would at least be bringing in something genetically similar. That's not to say they do very well. The environment's not great for them today, but one could consider these options. And as I said, one could consider boosting Indian lions, who are really in a very, very bad way, with West African stock, because they're not that far away and the admixture was happening anyway. So anyway, to end now, it's just some basic thoughts. What I really wanted to just show that was, once you do have good reference genomes, and more and more and more of these are coming all the time, with temporal data sets, we can actually identify genomic changes, and we can then decide if they were linked to the Anthropocene or not. We can also look at both diversity changes, but I didn't go into it here. Also, you can start looking at evidence of selection. When you've got stratified populations through time, there are some very nice tools to look at when does selection kick in, and therefore what is exactly driving that right now. And this is the kind of information that might be useful in helping prioritize conservation. I will say that I'm basically a pessimist and don't believe that will happen, because I think there's too much politics in conservation and people don't necessarily conserve things for the right arguments. But at least we could do it if we really got our act together. And ultimately, going back to my first point, maybe this will help get back to the question of, is this focus on genetic diversity the absolute best thing right now? What situations is it important? What situations is it less important? Maybe, for example, the Channel Island foxes are fine because they're so isolated, there's no threats, but maybe something with similar low diversity elsewhere is in trouble because of other reasons. But we can't make these decisions until we have the data, and to have the data, we need the temporal stuff. And therefore, it's great that Mirav's got a lab working in your museum. So thank you. Thanks very much to Mirav for inviting me to talk. It's always a pleasure. I'm sorry I couldn't come in person, but there you go. You'd have all got sick from the Danish COVID. Of course, many collaborators are involved in this, but mainly thank you to the audience for listening. And I will hang up here.
Thank you very, very much, Tom, for this great talk. Thank you. And now our second speaker, I would like to introduce Professor Ian Barnes from the Natural History Museum, London. Ian will talk about the changing structure of the prehistoric British population during the introduction of agriculture. Ian, please. Thank you.
Okay, thanks, Maria. So I'm just going to share screen now. And go to presenter view. Does that look okay? Yeah? Yeah. Okay. Also, thank you so much for inviting me to come along and speak today. I'm really pleased to see the lab and all the work that Marav's been able to do previously, and you know, it just looks like there's going to be some really nice stuff coming out of the new lab. And I'm really looking forward to coming and seeing it in person as well, and maybe getting in there and doing some lab work with you, Marav. Just kidding, obviously that I'm not going in the lab at all. I'm very grateful for the opportunity to speak today, but it did put me in a slightly awkward position because actually most of the work that I've been doing for the last few years has either been on DNA preservation in museum collections, or it's been on collections genomics in conservation. So knowing that Tom and Matthew were speaking, and they're kind of world authorities on that subject, I had to kind of fish around slightly to find something to talk about. What I would, so I'm going to sort of introduce where I work first. The Natural History Museum building was opened in 1881, but the collections themselves are much older. They go back to probably about a century earlier than that. There are around 80 million objects in the collection. Around half of those are insects. And it probably has the world's largest collection of type specimens. We also have a lot of human remains. You'll notice that these numbers are always a little bit fuzzy because the collection is so large and extends back so far. So we think there are around 30,000 human remains from various places around the world. And I'm going to talk about human remains today, but I'm just going to drop in the other thing that I also have been spending a lot of my time, most of my time on, for the last couple of years, is this project, which isn't an ancient DNA project, Darwin Tree of Life, which is an attempt to sequence all the UK plants and animals. So every species, plant and animal in the UK, there are around 70,000. We have some money from the Wellcome Trust to do the first couple of thousand genomes, and the museum is playing quite a key role in that, and I'm the lead person at the museum. So I would actually like to talk about this because of the point that Tom made right at the end of his talk, the importance of reference genomes. So reference genomes are absolutely critical for doing ancient DNA work, and they've really hampered my ability to do cool projects with the museum collection in the past. So having these reference genomes is going to be absolutely central to it. But actually, at the moment, there aren't many outcomes from this project. I could have given you an interesting talk about the Samples Working Group Manifest Subcommittee that I've been chairing for some time now, which has determined the minimum number of fields required to describe a field collected specimen and make it suitable for genome sequencing. So we wouldn't believe how long that's taken, how many people that's taken to get that Excel spreadsheet set up. The answer is actually 58 fields, but I think we've just added 2 more actually. So a lot of the work at the moment has been setting up those kind of activities and setting up the structures that we work in, and it would be interesting to actually talk about that later once we actually have some real outcomes from the project. And although it's UK plants and animals, about half of global families, taxonomic families, are present in the UK. So the utility of these data will be sort of generally globally applicable. And I just wanted to throw in here, obviously, that we're a bit off schedule because of the problems of that we've had in the last year for doing field collection with COVID. So lots of lone working or socially distanced working to try and collect the specimens. And I'm really looking forward in future to trying to explain what's going on in that right-hand slide to somebody. Why are people wearing face masks in the middle of a forest? It's just not going to make any sense in like 30 years' time, is it? Okay, so that's the Tree of Life project. And it's important because of the relevance of reference genomes. About 10 years ago, it really is, when I've thought about it, it's going back that far. I started this project with, started talking about doing a project with Mark Thomas at UCL to look at human history, human prehistory and history in the UK. And part of the driver for that was that at that time, there was one really good reference genome, which was the human genome. And lots of population level data for humans. And that was the case for humans and not really the case for most of the other organisms that we were interested in working on. So we put a project together based around those kind of constraints to explore the history of the UK through ancient DNA sampling. We ended up being funded by the Wellcome Trust. We looked at over 300 individuals with a long time span and a big geographic range, at least big within the UK, which is not exactly a big place. And we put a bit of an emphasis on the older specimens, which is kind of what I'm going to be talking about today. There are some practical limitations, some practical issues for working with archaeological material of this age. So I'm going to talk about those just for 4 or 5 slides, and then we're going to talk about the case study, which is the introduction of agriculture. So DNA from archaeological specimens has some particular properties. It is short in length, and that very much limits the kind of technologies that you can bring to bear to it, and it makes working with it a specialist occupation, which is good for people like me and Merav, because it gives us a specialist job to do, but not so good actually for actually getting useful data out. So it's short. It's generally present from archaeological specimens, at least. It is found with a large amount of other DNA present as well. Much of that seems to be bacteria. Some of it is fungal, some of it is human contamination from the environment. We also tend to find that there's relatively small amounts of it there. So there's little information for the amount of sequencing effort that you put in. So we call that the complexity. And it means that for any given base position in the genome, there may only be 1 or 2 fragments covering that position. And so there are various get-arounds for working with DNA of this type. And I've already mentioned how important it is to have a reference genome. You have to pick your samples carefully, and that is made a lot easier if there are many, many samples to choose from. And in the case of the UK, for British archaeological material, it's, there's plenty of that. It's been well collected and well researched over the years. And then there aren't many get-arounds for that complexity issue, but there are a few technical ways you can deal with it. But that reference genome and having plenty of specimens are important points to make here. And so just flagging up as well the relevance of the read length distribution. How long does it take? How quickly does DNA actually break down? And that's a question that Matthew Collins and I have been really quite interested in for many years now. Probably one of the first conversations I remember having with Matthew was on this topic. And it now looks like DNA seems to decay very rapidly after the death of the organism and then kind of settles down somewhat after that. So again, that means that the kind of methods that we've been using to look at our archaeological humans are actually highly relevant to everything in the museum collection, unless it's something that's already just went immediately into a freezer. So here, for example, both the 5,500-year-old human and the 30-year-old archival wasp seem to have DNA fragment lengths of around 55 base pairs. And so most of the degradation that we see seems to occur shortly after death. So I mentioned fragment length before. I also mentioned the endogenous content. So how much of the DNA we recover is actually from the host, the original host. It's been for some years now, we've known that some bones are better than others for recovering DNA from. And this figure is just flagging up the results from our 300 or so British archaeological humans. It seems that the petrous bone, the bone in the inner ear of the skull, has much higher endogenous content. So there's much better preservation of human DNA in there and less invasion from soil bacteria. So around a quarter of the petrous bones that we looked at had more than 20% human DNA content, and the figures are much, much lower for other elements. I'm still trying to work out what determines the non-endogenous content. So why do some samples have some sets of bacteria? How much variation is there in the bacteria that they picked up? Are they picking up bacteria and fungi when they're actually stored in a museum? It's very difficult to pick out patterns here, and I suspect it's because there's so much data that it's difficult to kind of break that, or to kind of combine that data together in the correct way. And the only pattern that seems to be dropping out at the moment is that material from river deposits does seem to cluster together. You can see it on this principal components analysis down in the bottom left-hand corner. It's the kind of yellow dots with the word river next to them. So for the most part, though, there's just seems to be quite a large degree of variation in the invasive community that occupies the body after death. So a lot of the data is large and complicated and difficult to work with. Sometimes, though, we clearly benefit from having these large data sets, and here's a flip side to that. So lots of people have looked at whether, not lots, but a few people have looked at whether there's a relationship between the amount of mitochondrial DNA and the amount of nuclear DNA that we recover from archaeological bones. And previously, it seemed that there wasn't much of a pattern, that you could have good preservation of mitochondria and poor preservation of nuclear DNA. Here's the results from the run that we did, or the work that we did. And I think the difference here, because you can see there's a very clear correlation, but the difference I suspect is because these data all come from a single individual doing all of the laboratory work. My colleague Selena Brace, who was working on the project, using pretty much the same method all the way through in the same place with samples from a relatively pretty much the same area. The UK is not very large. So you can see here there's a very strong correlation between nuclear and mitochondrial DNA content, and that suggests that those two different genomes are degrading at the same rate pretty much. You'll also notice, if you're very good at picking out different colors, because the key on the right actually hasn't got much color variation, but you will notice that the petrous bone samples always seem to fall above the line, and the other DNA from the other elements falls below that line, and that suggests that for a given amount of mitochondrial DNA, there's relatively more nuclear DNA in the petrous bone. Okay. So those are the kind of technical limitations that we are dealing with. Short fragments, lots of other DNA, other junk DNA that we don't really have much interest in, and a limited complexity, so low concentrations of DNA that make it difficult to actually identify exactly what's going on, limited information content. Okay, so on the flip side, the archaeological, that's the kind of chemical, technical limitations, but here's the interesting bit, the archaeological setting. So we had samples for that Wellcome Trust funded project that stretched all the way across this time period, and actually even further, more recently, into the sort of early medieval period. But the number of samples that we were able to obtain from different time periods was quite varied, and particularly for the Mesolithic period in the UK, there's very little material. So I think we ended up, through the very diligent and clever work of my then postdoc Tom Booth, in sampling pretty much every Mesolithic individual that existed in the UK at that time point. And most of those didn't really yield a lot of data. The point we're interested in here that we're going to talk about here is the transition to farming, which happens around 6,000 years ago. It happens very late in the UK, and the reasons for that are unclear. I should mention that this work's now been published. So the arrival of farming in Britain has been the subject of discussion for over a century. I did it as an undergraduate on several different lectures. I can remember talking through this, and there was still quite a heated debate when we started working on the paper. So the two big questions, really the big question, is the nature of the process. Farming, as I say, arrives late in Britain. It gets all the way through Europe and then seems to hang around for a number of centuries, I think actually pretty much almost 1,000 years in sort of northwest Europe before jumping across the Channel and getting into the UK. So is that because of human migration? There are perhaps limits to human migration, and then finally humans migrate into Britain, bringing farming with them? Or is it because the idea of farming enters into Britain via a process that we refer to as cultural diffusion? So farming itself transfers over, but the people stay the same. That's a very tractable problem to address if we are looking at, if we can get some ancient DNA genome data, population replacement or continuity. And if there is a migration event, exactly where do those people come from? And various proposals have been made by that, are made on that point by Alison Sheridan, for example, and various other people. So as I said, it's relatively straightforward to obtain Neolithic, early Neolithic samples from the UK. There are lots of sources of material, a number of which are in the NHM collection, and others dotted around the rest of the UK. Mesolithic samples are much more difficult to find. Many of them come from southwest England. You can see there's this kind of congregation of green dots, which represent the Mesolithic samples we got working, that are from southwest England and also southern Wales. And one individual from Cnoc on the west coast of Scotland. These are, we generated genome-wide data sets, tens of thousands to millions of single nucleotide polymorphisms, so individual pieces of information for each sample. And we also sequence up to proper genome size coverage for a couple of individuals. So one was at 2.3x and one was at 10.7x coverage of the genomes. And just for those of you who are not I realise it's quite possible that you may not be that familiar with national geography in the UK. So Scotland's the bit in the north, Wales is the bit on the west-hand side, England's on the east-hand side. So how different were the people from the British Mesolithic to the first farmers? I mean, this is basically our core question. Is there a population replacement? And the answer is yes, there absolutely is. So that's that one ticked off, really. So the British Neolithic samples on this principal components analysis fall kind of pretty much in the middle of the plot. They're the black shapes, and the Mesolithic individuals are the green shapes that are on the kind of central left-hand side. So there's very clearly a major difference between those two population groups. British Mesolithic individuals cluster with Mesolithic individuals from elsewhere in Europe, other Western hunter-gatherer groups in Europe. The British Neolithic ones are clearly part of a general expansion across Europe of Neolithic farmers that begins some thousands of years earlier. Who are the British Mesolithic individuals most similar to? Well, they're part of this Western hunter-gatherer group, and actually, if we drill down beyond just the simple principal components analysis, we can find that the Cheddar Man individual, so one of those individuals from southwest England, is most closely related to an individual from Lochbourg in Luxembourg, and also pretty much indistinguishable from an individual from Ranchot in kind of central East France. So it seems that there's a kind of a single population that extends across Western Europe and into Britain. And other data we've subsequently generated suggests that that group must arrive in the UK around 14,000 years ago. So given that there is complete replacement, it seems, a very significant degree of replacement between the Mesolithic and the Neolithic, also indicating that farming arrives via a migration event. Where do those Neolithic farmers actually come from? We can use here F4 statistical analyses to look at how much sharing, haplotype sharing there is between our test populations and certain kind of control populations from different places in Europe. So two plausible sources for those Neolithic individuals would be Central Europe, then spreading up into Belgium and perhaps across the Channel that way, or Iberia and spreading up either around the coast of France or across France and crossing over the Channel that way. And the data seem to pretty strongly suggest an Iberian origin, or at least that our UK samples from the early Neolithic are closest to Iberian specimens. So when those farmers arrive in the UK, having moved somehow from Iberia up into across the Channel into Britain, do they mix? Do they admix with British hunter-gatherers, or do they stay pretty much distinct from those? So there's some possibility, it seems, of admixture with British hunter-gatherers, but it's clearly not very much. And this rather confusing kind of structured figure here, it shows the data for that. Draw your attention to the fourth coloured bar down, the one that says Wales EN, so the Welsh early Neolithic individuals, if we consider them to be a combination of Western hunter-gatherers, so Mesolithic people, and Iberian early Neolithic people, what's the balance of those two genome sources? And the Wales early Neolithic ones can be explained as being exactly like 100% Iberian early Neolithic. But other places in the UK, there seems to be possibly some additional Mesolithic input, Western hunter-gatherer input, but it's relatively small. And so the data strongly suggests that if the origin is Iberia, there's almost no mixing of those populations by the time they arrive in the UK. And we can actually go through the data to try and look for possible introgression, possible admixture events. And we actually find that it's possible that there are two individuals from a cave site in Western Scotland that were the result of an introgression between Neolithic and Mesolithic individuals that occurred, in their case, about 4 generations before they lived. It's sufficiently recent in their past that it probably occurred in Britain. So it didn't occur before they got there. That's 2 individuals out of the fairly large Neolithic data set that we accumulated. One of the other things that we can look at is there does seem to be some degree of spatial variation in the amount of hunter-gatherer ancestry. It seems perhaps to be higher up in the northwest, Scottish Highlands, and lower in the southwest, so in the Cornwall region. And so if those aren't due to local introgression with Western hunter-gatherers, with Mesolithic people, could it be possible that these represent different source populations arriving into Britain? And so we tentatively suggest that in the paper, that this may indicate 2 different points of entry. And actually that goes back to some of the other evidence from the archaeological record, that perhaps there is 2 different colonizations of the UK, one into the southwest, that's a slightly earlier one, and a second one that's into the northeast, that's the later one. So in conclusion then, well, so the overall conclusion from that's specific to this talk, specific to this data set, the Mesolithic Neolithic cultural transition is driven not by a diffusion of ideas, but by a near total population replacement in Britain at that time point. The incoming populations seem to have come probably originally from Iberia. They come through France or around France in some way, and maybe enter Britain from 2 different locations. More generally, so what I wanted to flag up, and it kind of ties in with what Tom was saying earlier, ancient DNA methods that we use can be applied to a wide variety of different ages of material. So it doesn't, ancient DNA isn't necessarily old, it's poorly preserved. And one of the things that is really clear looking at lots of different bits of museum collection is that it's that DNA fragment length change, which is the really important one. So there's other aspects of DNA damage as well that are relevant, but it's the fragmentation and how you analyze those data, how you recover the short fragments and how you analyze them that are really relevant here. I'd also just mention as well, once again, resolving those kind of population level processes using DNA from museum collections is made a lot easier if you have a high quality reference genome. It also helps to have large modern data sets and lots of material to work from as well. So obviously you can't always fit your project to that set of criteria, but if you're weighing up different possible projects, I would say that this is important to bear in mind. Okay, and I think that's the end. Thanks very much.
Thank you, Ian. Thank you very, very much for a really interesting talk. I forgot to say that if you have any question, you can just write it down in the QA box, and we will, in the end, we will read them through. And now for our last speaker, I would like to introduce Professor Matthew Collins from University of Copenhagen and University of Cambridge. And he will speak about what is useless and what is useful about ancient proteins. Matthew, thank you.
Thanks very much, Marev. And yeah, I always complain because unfortunately, Marev didn't do a PhD with me. She went to work with Ian. Never forgive her or him for that. But nevertheless, congratulations. It's wonderful to see the new lab working. And I think it's also wonderful to see the fact that you're essentially trying to democratize DNA. So you're making these methodologies available to this much wider group of researchers. So yeah, what I want to talk about is kind of like overview where we are now with ancient proteins, because I think it's particularly actually relevant when we're coming to places where we've heard a lot about, high latitude sites. So we've heard about mammoths, we've heard about the UK, but when we come down to warmer sites, we know things are much more difficult. And that's a point at which proteins can potentially have more of a role to play. And so what I want to talk about then is, what is paleoproteomics, very briefly, and then when it's useless, then when it might be useful, and then where we might want to go from there. So basically, for me, when we talk about paleoproteomics, and it's a word that's been thrown around in the last few years, what it essentially means is getting enough information from the ordering of the individual amino acid residues so that we actually can identify what a particular protein was. And there are therefore materials which have not obviously been stored or preserved in such a way to preserve proteins. So it's a very simple definition, and there's a range of different technologies that we can use to get out the sequence information. But the sequence information is only ultimately useful if we can then use that to pin down the origin, the type of protein, and where that protein has come from. And I would like to remind you, we've heard a lot about DNA, but of course, we are essentially bags of water on most living organisms. And yet the next most dominant components are proteins and fats. And so if we're going to be dealing with any kind of material culture, which is made of organisms in one form or another, plants or animals, this is animals, this data here, then it's very likely that we're going to leave behind traces of proteins. So we're working with bone tools or pigments or glues or gums, food residues, there's going to be proteins preserved. And so what I want to very, very briefly mention is how we can do the analysis. Well, essentially, we'll take our object and we'll extract the proteins from it. And one of the tricks that we've been exploiting in the archaeological records is that many of the proteins don't survive well. So if you take an object like bone, essentially it gets purified over time so that it's only a single protein or a dominant single protein, collagen. And it's therefore quite easy to extract that. And in the early world of proteomics, what would be done would be to separate out the proteins usually in 2 dimensions on gels, by charge and by size, and then take a single spot which represents a single protein, and then you could fingerprint that. So the idea of fingerprinting a protein is quite common. And the way you fingerprint is you take the protein, then you cut it into small bits, and then you analyze it on what we call MS1, single mass spectrometer, looking at the range of sizes. Now, that is very fast and very cheap if the sample has been pre-purified for you, which is the case pretty much with all mineralized tissue, which is made with collagen and also, in fact, things like parchment. And that means we can use very rapid technologies, and we call one zooms, for very rapid low-cost species identification. As we get more sophisticated, though, and as we start to look at more complex mixtures, then we have to do a bit more. And so what we have to do then is we have to take a mixture of these peptides from multiple protein sources. So the peptides are coming from different places. We're going to separate them out using liquid chromatography typically. And then we're going to analyze the masses of all of those peaks. But it's not enough because we don't know where they came from. The single mass is no longer unique because it could have come from multiple sources. We have to do something else to it. Then we have to take those individual masses and we have to smash them apart. And we do that usually by flying them through a cloud of gas in a vacuum, and they smash into the gas atoms and fall apart. And then we can look at the fragments, and that's the MS2 analysis. So now we're going from an MS1 to an MS2, and we're using liquid chromatography. So we hear the terminology LC-MS-MS to indicate the kind of analysis. And then the real challenge, though, is that in that kind of analysis, we talk about protein sequencing, but it's not like DNA sequencing. It's actually much more pattern matching. And because it's pattern matching rather than direct sequencing, you only find what you're looking for. And this is a real problem when it comes to archaeology, because if we're looking, let's say, at a complex mixture of microorganisms in dental calculus, or maybe looking at a mixture of foodstuffs that come, you know, from a cooked mixture, we don't know what might be there. And so we don't really know what we're looking for. And so we have to then use very large databases and theoretically cut up all the peptides we might generate and then try and match them. And that gets more difficult still, because over archaeological time, we get further modification and damage. And so we have to account for all the possible changes. And the scary reality is, at the moment, we are pretty useless. And we're useless because we know that of the maybe many thousands of queries that we get in a single run, we can typically, in an ancient sample, identify about 3 to 5% of them. So that means that the remaining 97 to 95% of those queries that we've generated, we have been unable to pattern match, even with quite sophisticated technologies. And so it's telling us that we are building up a very large data set of dark matter proteomics, which will actually need additional interrogation. Unfortunately, now most researchers put all their data raw online. So when we actually are able in the future to understand more of what we see present, we can identify it further. But it is a pattern matching problem. And the other problem is then that because of that, we only get probabilistic identification. And in modern proteomics, maybe less so in ancient proteomics, we have the thing called a one-hit wonder. We generally don't want to say we've got a protein when we only have a single peptide. We need to match it up to multiple peptides. And another problem then arises because if we have multiple peptides which could derive from one protein, there's a very large degree of homology between these things, then even if we get an additional peptide from another protein, we all, by Occam's razor, say it probably came from protein 1. So we will probably underestimate the diversity of proteins we find as well. And that gets worse when we're finding fewer and fewer peptides. Now, the most important thing to realize is this scale difference. DNA is vastly more informative, as you've heard from Ian and you've heard from Tom. The difference is, though, DNA is a much larger number of atoms. And so here we have a strand of double-stranded DNA. And what we've done, I've shown the atoms of the DNA, and I've tried to scale these as accurately as I can. And then we've shown the peptide that those DNA sequences actually make. So all of these molecules here are making this package of information here, so that you've got as much information coded in this little small molecule as you have in this long string of large molecules. So whilst unfortunately only a very small fraction of the total DNA is actually coded in proteins, those messages are small, they're compact, they have fewer atoms in it, and many of the atoms are atoms which form bonds which are much more stable. So we have those carbon-hydrogen, carbon-carbon, carbon-nitrogen bonds, rather than think phosphorus-oxygen bonds. And as a consequence, these things will persist generally better in the archaeological record. But as you heard from Ian, our understanding of how DNA survives is still rather poor. And in fact, we are, with Lover-Dahlen now reaching back more than 1 million years, DNA is remarkably robust too. So now I want to talk about useless, and this is, I don't by useless bad. What I mean about is the learning experience we're currently going through. We are many years behind where DNA is, and the community is smaller, so the progress is more it's more tardy. And what I want to talk about then, this is actually coming from one of those motivational slides about mistakes. And there's different kinds of mistakes. And there's mistakes where you make a mistake and you suddenly realize, I'm not going to make that mistake again. There's mistakes where you're just reaching a little bit too far. And there's some mistakes where you take a really big risk. And they're all kind of good mistakes. The only kind of mistakes which are bad mistakes are these kind of sloppy mistakes where we make silly errors. But because these new technologies are so new, and because most of the people working these new technologies are fairly unfamiliar with applying them to dirty archaeological samples, then mistakes can get made. And so I'm going to illustrate now a kind of sloppy kind of error that you can sometimes find. And this is a great paper. This is on finding brucellosis in milk from the Egyptian pyramids. And so they found here this wonderful vessel with milk in it. And then when they did proteomics analysis on this, it's well preserved. They find a range of proteins. So we have a nice range here of milk proteins. In addition, though, we find this particular protein here, highlighted here in pink. And this is a sequence which is related, which is found in brucellosis. And it's, remember, a single sequence. And so one of the things you really must do with ancient proteins, and really wasn't, I think it was done, but they decided to tell the story in this way, I'm not sure, because they knew that brucellosis was present in these populations based on what they believe were historical accounts. They then said it's brucellosis, but if you just take that sequence and just do a conventional blast search, this is the sequence and it's got 100% identity. You can go on and on and on and on. This is a highly conserved piece of sequence. And remember, one peptide does not a protein make. So they chose to interpret this to make a good story. And you have to therefore look carefully at the results. And very frustratingly with this study, they didn't publish the raw data. Remember, with us in proteomics, we are so bad at actually interpreting the data at the moment. It's really, I think, essential that we publish raw data so that in 10 years' time, you can go back and reanalyze this data. Another example, which is slightly less guilty, they definitely found the protein here, but a really remarkable story is where they found presence of the Crimean Congo hemorrhagic virus. So this is a less sloppy, it's a more brave mistake. I think I'm going to rescore it. But what happened here is what they'd overlooked was the protein they found was very widely used in the service laboratories and in the research facility in which they're working. So there's the research facilities using it, actually making this protein. And then there was obviously sequencing in their proteomics lab. An INH pot goes into the same lab and they find the same thing. But rather than looking for the obvious source where it might be, they come up with a very exotic story about a virus which is not present in Iron Age Germany, but perhaps the body got carried from somewhere further south where the virus was present. Probably the virus came from the lab. So that was kind of like, they were stretching. It was a great idea. They definitely found the sequence. They found lots of the sequence. They found too much of the sequence. And therefore, as a consequence, they probably should have looked in their own backyard first. And then there's another examples like this, and I think there's some really interesting places to look. So this is a bridge in the Czech Republic. And what this team have done is they've actually analyzed the mortar in this bridge. And that's, mortar is inorganic, but of course, sometimes you add modifiers to mortars, and that can often be animal fluids, blood and milk. And they analyzed this particular bridge in 2016, and they found a very, very few peptides associated with milk. And this then is published to look at the technology of the construction of the bridge and the use, in this case, of cattle's milk. We can tell it's from cattle. But the peptides are so few, and we were very badly caught out with a study where we thought we'd find milk in the pyramids in the cement used to pull the pyramids of Giza together, realizing ultimately it came from contamination. And I strongly suspect these results are so similar to ours. And yeah, again, the published data is not available, so you can't reanalyze the data yourself. This is probably, it may be right, but I think they probably reached too far, and it's probably contamination. And the reason for that is that if you go into Maria's prep lab rather than the ADNA lab, then there'll be lots and lots of reagents, and these reagents are sometimes they're derived from different organisms. Some of those reagents actually have additional things added, proteins added to antibody solutions, for instance, to stabilize them. And of course, you're using the common proteins. The common proteins are the proteins from the agricultural industry. So the things that we really care about, proteins, things like foodstuffs like egg and milk and meat and serums and things, unfortunately, they're also common in the laboratory. And so there's always a concern with proteomics that you're going to find those kind. And here's this really interesting paper that's come out of Israel. And I'm not sure about it. This one needs more work because there's very remarkable claims. Particularly, I think I'm intrigued by the banana, the musa here. But this was a single peptide. And so, yeah, it's really interesting. But, it kind of, needs more work. And I think what we obviously need to be doing here is looking at all the other lines of evidence. And of course, the problem with things like banana is we don't have a very good vegetal record. So you can see the power of this technology to actually look, in this case, at the proteins trapped in dental calculus, and actually look at those to look at the record of plant consumption. But at the same time, and this to be extremely careful, but at the same time, it's a really high wire act stuff when you're making claims which are really quite dramatic about very early use of plants in this part of the world. And this is another example, which is intriguing. I don't know if this is right or this is wrong, but this is a fascinating study. And it has, I think mostly it's right. But this is one of those kind of aha, and you start to think more deeply. So this is a study where that Shevin Wilkin has done a lovely piece of work analyzing dental calculus across Mongolia and has identified the presence of different animal species. And I think this is firstly a very important paper because early claims had argued, based upon lipid analysis, that horse milk was being consumed very early. And this big survey actually shows that horse milk is actually not exploited until much, much later. But the other thing which is really quite intriguing, if you go at the very, very earliest site, it's horse. Sorry, no, it's not horse, it's cow. And yet, when you look at the other evidence, it's much more sheep-goat environment. It's not cow, sheep-goat. And this has got us thinking. And so it's identified as cow based upon one peptide. I mean, there are many other peptides that support the idea there's milk present in the calculus. And what's intriguing about that is in sheep, we have one amino acid. In cow, we have a second amino acid. And in goat, we have a third amino acid. And that's fine. But going back to one of the things that was mentioned by Ian, I'm obsessed with diagenesis. And this is kind of one of these aha moments, because this amino acid will decay to that amino acid. So potentially, as you go back further in time, effectively a sheep can become a cow. And I don't think that's the case in this study, but that's led us on to start thinking really more about, can we begin to pull apart these mechanisms? And if we take that particular protein and put it in solution and cook it up, here's that protein. And so the first interesting aha component is actually this peptide that is used and found wide in the archaeological record is actually not surviving very well when the protein is in solution. And so what we've then been doing is we've been starting to use alternative approaches based upon trying to simulate these things computationally. So we've been using molecular dynamic simulations to start to look at that peptide, look at the dynamics of that peptide and how it performs, and then trying to link that data back to our understanding of survival. And what we're seeing here, for instance, is if we look at the mobility, we have this very, very mobile glutamic acid and rather immobile valine residue there. We can see differences between cow and sheep. And then we can start looking at the role that cations have to play. And so what we're doing is we're playing around by putting different cations into the system. And what we're seeing is that the glutamic acids are being locked in place by the presence of cations in solution, whereas we're seeing much less stability in the aspartic acid residues. So we can start to sort of piece together what's going on. And now what we're doing, and this is still very early work, is we're now putting these peptides onto mineral surfaces and probing them with our colleagues at the Globe Institute. And so we're trying to get a sense of actually what is happening to these peptides over archaeological time, and why it is that a peptide which is not surviving well in solution is the peptide which is surviving so far back in the archaeological record. And our previous work on eggshell would imply that this is a mineral organic interaction, which we think is going to be significant in this case, but it's a work in progress. So we're in the early days of all these studies. We're getting lots of interesting findings. We are missing huge amounts of data. 95, 97% of the data that's being generated, we can't interpret. I would say in a modern tissue sample, it's about 50%. So it's not that in modern samples they can identify everything, but nevertheless, it's a significant amount of missed information. So kind of where is it useful then? Well, I think the most obviously useful, the one that's had the widest application is this methodology of zooms, which is taking that MS1 analysis and just relying on the fact that proteins in these ancient bone samples are largely collagen. So we mainly get collagen. Well, as a consequence of that, it's used to screen lots of samples. Unfortunately, actually, in the Levant, even zooms, if you go back to these early PPMB sites, is actually working rather badly, but we're working on a paper on that at the moment with a new little technology trick that actually is, we've managed to finally get some results from some very hot areas, so that will be coming out soon. But what is really impressive about this is the people like Mike Buckley here who've just taken this on and just done thousands of samples. You saw the picture of Denisa, that was Sam Brown's work, and she did again thousands of samples of bone chips at Denisa Cave till they uncovered the human remains. And I think what's exciting about this is it does mean that in museums where there are lots of unidentified bits of faunal material, zooms can actually find things which are essentially morphologically useless because they don't carry any morphological information, and then find those things which could then subsequently be used for more destructive analysis for DNA, for radiocarbon dating, etc. So I think that's a real value of zooms. And in this study Mike did on this site in Yorkshire, in Creswell Crag, sorry, in Derbyshire, he was able to identify over 12,000 bone fragments and compare the zooarchaeological evidence from the bones themselves with the zooarchaeological evidence from the unidentified fragments. And in this case, they broadly agreed. However, in this case study by Frieda Welker and his student, Valerie, what they found was that in this particular case site in northern Italy, the zooarchaeologists were seeing primarily cervid remains, whereas when you actually looked at the very small fragmented bone remains, you're seeing a lot more bison. And so what they've argued in this case is actually what you're finding here is that there's been over processing of the bison, the large bone bison material. So it's missing in the zooarchaeological record, but it can be discovered using simple peptide mass fingerprinting. So you can see there's a value to that. And I think, to me, the most intriguing work which is coming out, I think there's going to be a lot more of this, is in the area of work bone. Now, the problem with work bone is it does require some form of destructive analysis. In the case of this particular study, it's historical. And so we're just able to analyze the bags in which the samples were carried. And this was looking at bone points, and there's now been a number of these. And in this particular set of bone points, what you're seeing is a site which is dominated by fish with some white-tailed deer. Actually, white-tailed deer aren't that commonly used as bone. Mainly we're seeing bear and human. And I was talking to one of my zoological colleagues that said that human apparently doesn't make very good work bone tools. I'm not sure how he knows that, but that's what he told me. So they're using material which is actually not that useful. But we do know about the cosmology of this group, and we do know about this relationship between bear and human-bear transformation. And so it's a study, it's a single point, n equals 1. This then really intrigued us. We did a very large study of Mesolithic bone points from Denmark, which told another story. It was actually a story about changing cultural groups using combination of zooms and radiocarbon, which I won't talk about. But this study then came out very subsequently to ours. Now, we had analyzed many bone points. They analyzed a relatively few from the North Sea. And the bone points with similar typology. So this is Mesolithic. In our case in Denmark, the points have been lost as they had been fishing, presumably, and they got stuck into the peat bogs. We don't know how these particular tools were lost. But if you notice, the two most highly worked of the 2 points here, and they're very small, and apparently they've been reworked, are again made of human. So now we have a historic site and a Mesolithic site, relatively few bone points, and yet significant numbers being made of human. And now I've just been testing to others who've also been finding the same thing. So this is now occurring, I think, from the Paleolithic right the way through, a selection for working on humans. And it seems strange to me because you're working on green bone, fresh bone. And if it's true that humans don't make particularly good tools, why so common? So I think using this protein ID of work bone, where you can sample very many samples very cheaply and quickly, is potentially going to be useful in the future. And Frieda Welker, one of my colleagues, is working on that. Since we're in Israel, I can't help but mention parchment because so many people are so interested, of course, in Dead Sea Scrolls. This is a project which is underway at the moment. And this is the most important book in the university's collection at the Ardemagnan Institute at the University of Copenhagen. And it's a thing called a palimpsest. So it's actually made of multiple different book objects, which have been scraped and then rewritten on. It's a hugely important text because it's actually it's the only copy of the Apocalypse, and it's a very, very early text from Spain. So what we've been doing is we've been helping out a team. They've been doing visual identification of this. It is their most important book, after all. It's going to be redigitized soon. We're hoping to do some multispectral imaging on it to get more evidence of the undertext to actually reveal the true answer. This estimate at the moment is based on rather limited paleography. What we've been able to do is just analyze the proteins with a simple eraser, soft eraser. You brush the surface, you generate electrostatic charge, you pull off proteins. And what we've done here is we basically grouped the proteomics data just by cluster analysis. And what you can see is we're starting to see clusters of similarity in goat in red here and sheep in yellow. And what's intriguing is, surprise, surprise, the different parchments have different qualities, which is related both to species, but also to production process, which is allowing us to see the different origins of the different groups of parchment. So we can begin to understand the link between the production and help guide the study of the underlying palimpsest. So what about populations? The confidence about populations, not so good on populations. The problem is when you're working on DNA, you have a whole genome to work with. A lot of it's non-coding. We're only working with the coding parts. And worse than that, we have to deal with the non-synonymous mutations, which are, of course, the ones that affect the DNA sequence and the ones which are most heavily selected against. So here we have a problem. So we're going to get much less information than you are going to get with proteins. But Frida Welke at Copenhagen is getting very interested in this for looking at human populations. And because we got so much human population genomic data, we can start to map, or him and his team with Pontus Skoglund and others can start to look for these mutations in the human genome and look for population structure. The other thing to, of course, be aware of is all of the substitution are not equal. So this is a map showing the patterns of substitution. And for instance, we can see that if you've got an aspartic acid, it's much more likely to be substituted by a glutamic acid, which is very similar chemically, and much less likely to be substituted by a leucine molecule, which is a hydrophobic amino acid. So, you know, not all substitutions are equal. And also beyond and above that, there are structural differences too. So it's much less informative. It gets worse than that because there's also positional dependencies. We'll get greater rates of substitution in loops than we will in helices and things like that. So what that means is that when you come to try and use collagens for sequencing, this is a study we did on Caribbean sloth. And what you see is that there's a huge amount of uncertainty. So this is using just collagen. It's not very helpful. And this is a really interesting study using all the proteins found in hair. Here they were able actually to demonstrate and discriminate hair from different large-scale population groups based upon these single amino acid polymorphisms. However, this I think was the most important, interesting part of this study. This is a plot showing the range of different proteins and their intensity shown here. It looks rather like a gel, but it's an illustration of the range of different proteins found in different hair samples. But they worked, fortunately, with an archaeologist to supply some archaeological hair, and we've done some work on this as well. And annoyingly, what you lose is you lose those proteins, which are the highly variable ones, more is the keratin-associated proteins, and you're left with the major structural proteins. They're the ones with the slowest rates of substitution, like in collagen. So unfortunately, this technique could not be applied to ancient populations because the bits of the protein that survive are the toughest parts are the structural parts, and they are the slowest to evolve. However, it can work. And this is work by Enrico Cappellini and Frida Welke from the Globe Institute. And they've been looking at enamel. Enamel is a really exciting substrate. And this is the first example of a kind of a really extraordinary ancient proteomic study of the few proteins trapped in enamel. And this was looking at Homo antecessor. Frida's done another one on the Gigantopithecus from China, and able to show here that antecessor is actually not ancestral to the group with modern humans. Neanderthals is actually sitting to the side of that. And what's exciting about this is we know that these proteins are surviving well, and also by a generous act from someone, the proteins themselves, the dominant protein, is coded on the X and the Y chromosome. So that also means that these proteins can be used for sexing. And this is a very famous recent case for the lovers of Medena, both of whom turned out from the enamel proteome to be male. Now, why is this exciting in Israel, it's exciting because these are really, really tough proteins. We're talking about proteins which will extend back multi-million years. And so here's an example. It's buried away in a paper looking at rhinos. But actually, this is a paper where they were able to sex these deer and identify these male deer in a sample from Dmanisi in Georgia. So this is a very old site where we're getting sexing, and we're very confident, or they're very confident, we will be able to go back significantly further in time. So sexing using enamel proteins is something which is going to become routine and is going to be very, very valuable. For me, it's most valuable. We did this work using DNA, looking at things like kill-off patterns in animal species. And so ourselves and others, many others, are now getting very interested in rapid ways of sexing bovid remains in particular, to look at kill-off patterns to identify sort of a daring strategy versus a meat strategy versus a mixed economy strategy. And that's something which is going to be very, very accessible in the coming years. And it's going to be particularly accessible by this development at Copenhagen, not by me, but by Enrico and others, Frida and Enrico, which is to try and go away from a Zooms type technology, which is relying on a single protein, to a high throughput automated LC-MS technology, which has very, very short runs. You can see here 2 to 4 minutes, but which gets not all of the proteins that you can find in a sample, but enough proteins in enamel is a very short proteome to enable us to do things like sexing. So what else can we do which is useful? Well, production is another area which I think, and I just throw this in, I think it's a wonderful study, a very old study actually by now. But this is a study where they found this, which is, of course, you all do this, right? You wear necklaces made of cheese. I mean, why not? Particularly hard cheese. And so this is this mummy in this very, very dry site in the Gobi Desert. And this team were able to identify the fact they had lactobacillus and milk protein. So we're talking about, that this is a fermented milk product, and they've got far more curd proteins than whey proteins. And when they then analyze the curd proteins, what is so neat about this to me is, of course, you can make cheese in different ways. You can use rennet, and it's a fascinating story of the adoption of rennet in the archaeological world. Who first, I guess, they saw a calf stomach with milk in it and realized it was curdled. But you can then, by pouring milk into a calf stomach, you can form precipitates of casein. But the thing is that it's cutting at one very specific site. So what this team did is they looked at feta cheese, which uses rennet, and compared it with this particular site. And in feta cheese, we get a cut here between a phenylalanine and methionine residue. All the feta cheese has that peptide cut at that point. It's not the case in this site. So in other words, from this 4,000-year-old group, they are actually fermenting using acid rather than using rennet. But I think that's so elegant that we're going to get a handle on production processes. We can also look at things like the age of the skin, and a number of us have published. This is a nice example from Enrico's team, looking at using the presence of fetal hemoglobin to identify young animals. So because, of course, these proteins are tissue specific, and they're on some of them are ontogenetically specific, we can identify the age of animals. We can then look at the quality, and this is looking at the damage patterns in parchment over time. And we're then looking at historical trends in the quality of parchment, just by measuring the protein data. And then this is some of the most exciting things. Again, I wanted to highlight this. This is Jesse Hendy's work from York. Lots of us are getting very interested in this, which is the idea of looking at food residues on crusts, and particularly on lime scale. And what's to me really exciting about this work is on this site, we can get the zooarchaeological evidence of animal exploitation, but then we can see in the lime scale, we get a range of dairy protein shown here in blue, meat protein shown in pink, and plant protein shown in green, grain in yellow. So we can identify different proteins from different sources, and they don't survive so well if you don't have the lime scale. And obviously lots of us are working on this at the moment. It's becoming more difficult rather than more easy, if I'm totally honest at the moment. And we're getting more and more, yet more dairy proteins are coming out, and less plant proteins than we'd hoped. But nevertheless, I think this is really exciting. And we get a sense of the use of particular vessels. And then the last thing, is one single slide, is I want to mention, of course, is the need to integrate. And actually there's been, because we've been running forward with the proteomics, up until now, there hasn't been enough real integration with lipid analysis, and with DNA analysis. This is one example where that was done. This is a very early paper on calculus, and you can see the real value actually of the DNA and the rather limited value, in fact, of the proteins, but how the two can work together, and one data set can offset the other data set. And I think this is going to be particularly important in the case of dental calculus when we're getting very exciting results about, for instance, like things like novel plants. So final slide, where are we going to go? Well, I mean, the great thing for us is we're just riding on the back of biotechnology. So there is improvements in extraction, there's greater automation of extraction. There's been a drive for sensitivity really coming from single cell sequencing. And this is a part of the world of personalized medicine, but that helps us. Sadly, at the moment, that seems to have rather plateaued out a bit. So for us, we'd like more sensitivity, but the problem with more sensitivity is means you do detect more contamination. Increased resolution, we'd love to get to the time when you actually can use proteins for stable isotope analysis, but talking to engineers, they think that's actually probably never going to happen unless you're doing a thing called top-down proteomics and you have large intact proteins to study. You can't do it from the short peptides that we work with. And top-down proteomics is now being used increasingly in cultural heritage because we can look at process modification and particularly cross-linking. Finally, we need to get a much better handle on what we call PTMs, or post-translational modifications. Remember, if we're only finding 3 to 5% of those queries, we've got to understand what the remaining queries are. Are they just poor quality data, or is there something we're missing? Clearly, as I've highlighted, the problem of contamination. You've got to be very clear what's been happening, been used in the lab that you're working with, what kind of samples they've been running. And it's a funny thing, contamination. Even if you get nice blanks, you then put a carrier protein in, it seems to drag contaminants out of the machine. So be very, very careful of contamination. And lastly, this is where we are working closely with DNA, extending those databases, because remember, we're not really sequencing, we're only pattern matching. So yes, thanks to an awful lot of people that have helped with this. And yeah, that's me. Thank you.
Thank you very much, Matthew, for this fascinating talk. I'm always amazed how this field is progressing so fast. So many things are happening. It's great. Thank you. And now I see that there are no questions. So I think this is the time to end this symposium. Thank you again for Tom, Ian, and Matthew for your time and for your sharing with us your experience and work. And thank you all for being here and listening. And I wish for some peaceful days here and everywhere. So please keep safe wherever you are. Okay.
I think we can echo that, and all the success with your new exciting journey.
Thank you. Thank you very much. All right.
See you soon, Mara.
See you soon. Thank you. Bye.